Welcome. Today we'll visualize the cell cycle and cell division in a way that helps you remember the steps for Class 11 biology. Think of the cell cycle as an ordered series of stages a cell passes through so it can grow, replicate its DNA, check for errors, and divide accurately. The cycle is divided into interphase and mphase. Interphase, which takes up about 95% of the time, includes G1, S, and G2. In G1, the cell grows and prepares. In S phase, the DNA is precisely replicated. And in G2, the cell readies the mitotic machinery. Cells can also exit into a quiescent G0 state where they stop cycling. However, for those moving forward, the M phase completes the process through karyokinesis, the precise division of the nucleus, followed by cytokinesis, the physical splitting of the cell body into two daughter cells. Mitosis itself unfolds in four main stages. During prophase, chromosomes condense and become visible. In metaphase, they line up at the equator of the cell. Anaphase is when sister chromatids are pulled apart toward opposite poles. Finally, in telophase, nuclear membranes reform around each set of chromosomes, setting the stage for cytokinesis and two daughter nuclei. Meiosis is a two-division process that produces gametes. Before meiosis begins, DNA is replicated so each chromosome consists of two sister chromatids. Meiosis I separates homologous chromosome pairs, halving the chromosome number, and meiosis II then separates sister chromatids, producing four genetically distinct haploid cells, the basis for sexual reproduction. Meiosis I has distinctive behavior. In metaphase I, homologous pairs or bivalents align as a double row across the metaphase plate, unlike the single file alignment in mitosis. During anaphase I, homologous chromosomes are pulled to opposite poles while sister chromatids stay attached, so the X-shaped structure is maintained. The result after telophase I and cytokinesis is a reduction from diploid to haploid ploidy. Prophase I is especially important because it's where pairing and recombination occur. It progresses through leptotene, condensation, zygotene, synapsis begins, pachytene, crossing over via the synaptonemal complex, diplotene, chiasmata become visible, and diakinesis, final condensation and preparation for metaphase I. Those crossover events physically exchange DNA between homologs and create genetic diversity. Meiosis II begins with two haploid cells produced by meiosis I, and both cells divide concurrently. Prophase II and metaphase II set up the chromosomes again, and in anaphase II, the centromeres finally split so sister chromatids separate and move to opposite poles. After telophase II and cytokinesis, you end up with four genetically unique haploid daughter cells. Cytokinesis differs between animal and plant cells because of their structures. Animal cells use a contractile ring to form a cleavage furrow that pinches the cell inward, a centripetal process. Plant cells build a new cell plate from Golgi-derived vesicles at the cell center that expands outward, forming a middle lamella between the two daughter cells, a centrifugal mechanism adapted to the rigid cell wall. To summarize the big contrast, mitosis occurs in somatic cells for growth and repair and yields two identical diploid daughters, while meiosis occurs in germ cells to make gametes and produces four variable haploid cells. Both processes are tightly regulated, so organisms can maintain chromosome number, ensure accurate inheritance, and generate the variation needed for evolution. That's it for today. Learn smarter with EduRev. Notes, test, and practice in one app. Download EduRev now on Play Store and iOS App Store and keep learning every day.